The role of aminoacyl-tRNA synthetase in prokaryotic translation is key to the success story of protein synthesis because this enzyme has the onus of ensuring that tRNA as an adapter molecule carries the correct amino acid. So let us look at the learning outcomes of this session and also understand how important the role of aminoacyl-tRNA synthetase is. So what are the learning outcomes? The tRNA, which acts as an adapter molecule, they are charged by or with an amino acid to its 3' adenosine. The adenosine is of that of the conserved CCA, and the linkage between the adenosine and the carboxyl group of the amino acid is a high energy acyl linkage. This is highly significant. This charging of the tRNA is carried out in two steps by an enzyme, aminoacyl-tRNA synthetase. Every amino acid has therefore its own dedicated aminoacyl-tRNA synthetase. There are two classes of aminoacyl-tRNA synthetases, and it is the enzyme specification to add the accurate amino acid to the cognate tRNA, as ribosomes are not able to discern whether the incoming tRNA has the correct amino acid. That means that ribosomes do not or are not able to discern or not able to distinguish whether the tRNA has brought a correct amino acid or not. So this step of adding correct amino acid to the tRNA is therefore completely the onus of aminoacyl-tRNA synthetase. They have several mechanisms of ensuring accurate charging of the tRNA. So when you say charging of tRNA or when you say charged tRNA, what does one understand? Say for example, you have just the tRNA, okay, without it being linked to the amino acid, then such a tRNA is what is called as the uncharged tRNA. Now, when the CCA at the 3' end of the tRNA, the A of that CCA is in an acyl bond with the carboxyl group of an amino acid, then the tRNA is considered to be a charged tRNA. So this acyl bond that is present between the adenosine of the tRNA and the carboxyl group of the amino acid, this is a very high energy acyl bond. And this being high energy is important while translation goes on, and when during the peptide bond formation, no external energy is required in the form of ATP, because it is this high energy bond that provides the necessary energy to synthesize or to form the peptide bond. Now, every tRNA, every charged tRNA has to be added with a cognate amino acid. Now, what do we mean by a cognate amino acid? Say, for example, the tRNA is sterile tRNA, which means that the sterile tRNA has an anticodon that is going to bind or hybridize with a codon specific for serine, which means that this tRNA will have to carry a serine amino acid. And therefore, for sterile tRNA, the cognate amino acid is going to be serine, which means therefore this accurate addition of amino acid to a tRNA is what is highly important. Who carries this out? It is aminoacyl-tRNA synthetases that carries this out. Now let us just look at the structure of the aminoacyl-tRNA synthetase. So as one can observe, the aminoacyl-tRNA synthetase has several different domains. So one of the most important domain of the aminoacyl-tRNA synthetase is what is called as a Rossmann fold, and this Rossmann fold, which is in red, is responsible for the catalysis or for the formation or for the charging of the tRNA. And hence, this catalytic domain is extremely important. As you can see, there is within the active site the binding of an amino acid. So an amino acid can come and lodge itself within the active site of the catalytic domain. Then you have what is in the blue is called as a connective peptide. As the connective peptide also has zinc fingers, and these zinc fingers enable this region to attach to the tRNA. Now you also have in magenta, as observed over here, another domain which is called as a stem contact fold. And last but not least is a bundle of alpha helices, and these bundle of alpha helices are specifically able to interact with the anticodon of the tRNA. So therefore this particular domain of the aminoacyl tRNA synthetase is what is called as anticodon binding domain. So you can already observe that the structure of the aminoacyl tRNA synthetase is such that it can bind both to the acceptor region of the acceptor arm of the tRNA as well as the anticodon domain of the tRNA. So if you can see, this is the tRNA, you can see that the acceptor arm, in fact the acceptor arm A is completely within the anticodon domain, and you can see the proximity of the amino acid that is present within the active site and the A adenosine of the acceptor arm of the tRNA, because the bond, the acyl bond that has to be formed, the reaction that has to be carried out by the enzyme is between the amino acid and the CCA of the tRNA. So therefore this is extremely important. And second, the binding of the anticodon to anticodon region to the anticodon binding domain is again very important. Now when one looks at the aminoacyl tRNA synthetase, it has been found out there are two classes of aminoacyl tRNA synthetases, class 1 and class 2. So it is known fact that at the 3' CCA end of the tRNA, the A that is present has both its 3' OH as well as its 2' OH free. Because it is an RNA molecule, it has sugar as ribose, and in ribose the 2' carbon of the sugar has OH. While the A being the last nucleotide of the tRNA will have its 3' OH free, because every RNA or every DNA has one end 5' phosphate and the other end is 3' OH. And so the A that is present at the 3' end will have its OH free, but it will also have its 2H free. Now big 2' OH free. Because you have the 3' OH and 2' OH both available, this OH that is present, these two OH that is present, can either or form the acyl bond with the amino acid. So that class of aminoacyl tRNA synthetase that attaches the amino acid to the 2' OH group of adenosine is the class 1 aminoacyl tRNA synthetase, while that class of aminoacyl tRNA synthetase that attaches the amino acid to the 3' OH group belongs to class 2. So the difference between the two, of course, structurally there are certain differences very evidently observed in this picture. But what is clear definitely is that both the class 1 and the class 2 aminoacyl tRNA synthetases clearly bind to the acceptor arm as well as to the anticodon arm of the tRNA. The only difference in the two being that one attaches the amino, so the charging of the tRNA is by attaching the amino acid to 2' OH, while class 2 attaches the amino acid to the 3' OH. Now let us look at what the catalytic action of aminoacyl tRNA synthetase is. In fact, the catalytic action is in two steps. So the first step is what is called as activation of the amino acid. As you can see in the figure, the enzyme has the catalytic domain's ability to bind to the amino acid. Now, however, the amino acid that is present inside is always in an activated form. Now, what do we mean by activated form? That is, the amino acid is adenylated. Now, what is adenylated? When amino acid reacts with ATP, within the active site of the aminoacyl-tRNA synthetase, the amino acid will link up through an acyl bond with the AMP. That means the carboxyl group of the amino acid will attach to the phosphoryl group of the adenosine monophosphate. And what is going to be released is PPI. We will look at it again as a reaction. The second step is the charging of the tRNA. So what happens is that as the enzyme is bound already to the adenylated amino acid, see, the enzyme is not going to leave it. In fact, the first substrate, that is the amino acid and the ATP, will remain bound to the enzyme as an enzyme substrate intermediate complex. This enzyme substrate intermediate complex has a conformation that allows it now to bind with great affinity to its cognate tRNA. So that tRNA is going to bind to the active site, and when it binds to the active site, the enzyme carries out its reaction in such a way that the adenylate group that is present with the amino acid is removed, while the amino acid itself is linked to the 3' end of the tRNA through the high energy acyl bond. So this is how the two-step reaction of catalysis happens with aminoacyl-tRNA synthetase. Just to look at it again, so the amino acid, its carboxyl group is able to interact with the alpha phosphate of the ATP molecule, thereby forming a linkage between the carboxyl group of the amino acid and the phosphoryl group of the uh, um, AMP, which means that this entire AMP moiety is going to bind to the amino acid at its carboxyl group. So the linkage of this linkage with this is leading to what is called as adenylation. If you say adenylation, then it is ADP being bound. But because it is AMP being bound, then it is adenylation. Okay. So therefore, the first step in the entire process is that an amino acid binds to an AMP. Because it is binding to an energy-rich molecule, it is called as activation of amino acid. So thereby, this adenylated amino acid is now present within the active site of the aminoacyl-tRNA synthetase. Post this, the incoming tRNA, which at its 3' end has the OH group that can act as a nucleophile, this nucleophile will again target the carboxyl group of the amino acid in such a way that this OH forms a ester bond with the carboxyl group, thereby getting an acyl bond, while the AMP is released. So you will see that the AMP is released, but you can very clearly observe that there is a storing of energy in the newly formed acyl bond. So here the bond that is formed between the tRNA and the amino acid, this bond is considered to be a very high energy bond. Like mentioned earlier, breaking of this bond releases energy, and the energy released due to breaking of this bond helps in the formation of the peptide bond in the ribosome during protein synthesis. So you can also look at the fact that this tRNA, other than working as an adapter molecule, is also storing energy for a putative bond formation during protein synthesis. Interestingly, if this reaction has to be carried forward, like in DNA polymerases, when PPI is released and it is converted to 2PI, the coupling of this conversion of pyrophosphate, inorganic pyrophosphate to 2PI leads to a delta G which is more negative, thereby making the reaction more favorable and moving in the forward direction. So the conversion of PPI to 2PI helps the entire process to move in a forward direction. Now, aminoacyl-tRNA synthetase therefore has two substrates. One is the amino acid and second is the tRNA. So, aminoacyl-tRNA synthetase has a big responsibility in ensuring that it is choosing the correct tRNA to attach the amino acid to. That is one. The second thing is that, say for example, you have an amino acid which can bind to 3 different tRNA. Why? Because say for example you have serine, and serine is coded by 6 codons. So the 6 codons can be recognized or can be bound to by 3 tRNA, which means we are talking about over here as there being 3 tRNA for one amino acid. So all the 3 tRNA that can bind to a single amino acid are called as isoacceptor tRNA. Now, there is only one dedicated aminoacyl tRNA synthetase for an amino acid. So if serine has to be attached to a tRNA, then it is aminoacyl tRNA synthetase, that is seryl tRNA synthetase, that can attach the serine to the tRNA. But it means it is that same seryl tRNA synthetase that has to bind serine to all the isoacceptor tRNAs. So therefore, what is the second challenge for aminoacyl tRNA synthetase? It not just has to choose the correct tRNA, but it should also be able to bind the serine to all the 3 isoacceptors or all the isoacceptor tRNA. So not just recognize the tRNA to which it has to bind the serine to, but also recognize all the tRNA that can bind to serine. So that is quite a job. So therefore the question arises here is, how does aminoacyl tRNA synthetase ensure this? So let's say for example you have the tRNA, and what is observed is that both the acceptor arm of the tRNA, especially when it is in its tertiary structure, and the anticodon region of the tRNA, both these regions are recognition specificity determinants. That means the aminoacyl tRNA synthetase is looking at interactions that it can make with both the acceptor arm and the anticodon arm, thereby ensuring that it recognizes the correct tRNA. So, at the acceptor arm, we know that every tRNA at its 3' end has CCA, and the CCA is a conserved sequence in all tRNA. That means that all tRNA synthetases in its or within its active site has a microenvironment that can interact with CCA. That is definitely the case. But what is interesting to note is that other than the CCA, the acceptor arm can also have other nucleotides which are enabling the enzyme to distinguish between one tRNA and the other. So such nucleotides which enable discerning the difference between two tRNA are what is called as discriminator nucleotide. If changes are made in these nucleotides, then what has been observed is that recognition of the tRNA by the aminoacyl tRNA synthetase is affected, and hence the tRNA being charged is also affected. So therefore this is an extremely important region for the recognizing of aminoacyl tRNA synthetase of its correct tRNA. Then within the anticodon region is present the anticodon stem. Of course, it also recognizes the anticodon itself, that is the 3 nucleotides that can hybridize with the codon on the mRNA. That definitely is one recognition point. Plus, you have the anticodon loop also with which the enzyme is able to interact. As observed in the structure of the aminoacyl tRNA synthetase, there is what is called as the domain 1 and domain 2 that can bind to anticodon region. And so this interaction again is very important. So all the three make very specific interactions with the anticodon domain of the aminoacyl tRNA synthetase. And this therefore is very crucial. Now, one would tend to believe that therefore the anticodon should be the discriminatory region to actually enable the aminoacyl tRNA synthetase to know which tRNA it is binding to. If here you have the anticodon for serine, then the aminoacyl-tRNA synthetase on binding to these three nucleotides will recognize it as a tRNA for serine. But like we mentioned, suppose there are three isoacceptor tRNAs for serine, then even by one nucleotide, you would have a change in the anticodon, which means that aminoacyl tRNA synthetase will have to recognize not one anticodon but a set of anticodons, and all the anticodons are for serine. So therefore, anticodon cannot be the sole discriminatory recognition determinant or specificity determinant. So there are these other regions, that is anticodon stem, anticodon loop, the discriminatory nucleotides, which help the aminoacyl tRNA synthetase to finally decide whether this is the correct tRNA or not. So these other recognition specificity determinants are considered to be the secret second code for the aminoacyl tRNA synthetase to recognize the correct tRNA. Now, let us look at the other substrate, which is amino acid. In fact, for aminoacyl tRNA synthetase, this is even more challenging for the simple reason that amino acids are small in size, and also all amino acids have common features with respect to the alpha carbon being attached to the amino and the carboxyl group. So what remains different in the amino acids is the R group. And based on the R group, the aminoacyl tRNA synthetase has to distinguish one amino acid from the other. So as we can see, every amino acid, this part of the amino acid is exactly the same in all amino acids. What is going to differ is the R group. Now, say for example you have glycine and say for example you have phenylalanine. Then structurally, glycine and phenylalanine are quite different with respect to the R group. Here you have hardly a bulky adduct, whereas here you have a bulky adduct. Here this is aromatic, here you don't have any aromatic. So in terms of properties also, the R group is different. Secondly, the size and the shape of the amino acid is going to vary because of the R group. So a glycyl tRNA synthetase can definitely be able to accommodate glycine but not accommodate phenylalanine because of the difference in the structure. Similarly, phenylalanine tRNA synthetase can accommodate phenylalanine because of its specific interaction with the aromatic ring, but would not be able to interact with glycine because it does not have an R group at all. So you can see that based on how the active site is able to interact with the amino acid decides whether you are going to have a favorable interaction or not, and that is one way of distinguishing one amino acid from the other. But that is because these two amino acids are very different from each other. Suppose now you have another amino acid, tyrosine, which is structurally very close to phenylalanine. The only difference in the two being that the aromatic ring or the benzene ring has in tyrosine an additional OH group. So for the tyrosyl tRNA synthetase and for the phenylalanine tRNA synthetase, till here the structure is the same. The only difference that is there is this OH group. So it becomes an even greater challenge for both the aminoacyl tRNA synthetase to distinguish one from the other. Now let us take another example. Say for example you have valine and you have isoleucine. What you observe over here is that the R group differs from each other only by one methylene group. This has one extra methylene group than this. Secondly, both are branched. So you can see that you have branched present, and of course both are again non-polar. So in terms of amino acids, you have very little difference in the structure, size and property of these two amino acids. So again for the aminoacyl tRNA synthetases, it is a challenge to distinguish between valine and isoleucine as well. Therefore, to enable the aminoacyl tRNA synthetase to distinguish between the two, there has to be a way out, and same way for here. Now let's consider, for example, the tyrosyl tRNA synthetase. The tyrosyl tRNA synthetase can very clearly distinguish between the phenylalanine and the tyrosine because of the presence of this OH group. The OH group in the tyrosine is able to make specific hydrogen bonding with amino acids in the active site that is going to favor the free energy change. And that free energy change makes the enzyme spontaneously bind to tyrosine but not favorably to phenylalanine. So the delta G negative that becomes because of the interaction of the amino acid to the enzyme active site is one of the ways by which amino acid specificity is determined by the aminoacyl tRNA synthetase. Now let us come to this. So we know that valine has this branch structure and the isoleucine has this branch structure. Now, the valine tRNA synthetase can easily distinguish between valine and isoleucine for the simple reason that the active site of valine will not be large enough or big enough to contain the larger R group of the isoleucine. So therefore, it can bind to valine more effectively, but it will space out or it will not allow the isoleucine to enter the active site. So that is how valine tRNA synthetase can easily choose valine over isoleucine. But what about isoleucyl tRNA synthetase? The isoleucyl tRNA synthetase has an active site that should be able to accommodate the R group of the isoleucine, and this R group of the isoleucine is bigger than the R group of the valine. So therefore, basically a valine can definitely enter the active site of the isoleucyl tRNA synthetase because it is smaller than the isoleucyl tRNA synthetase. So, one way by which the isoleucyl tRNA synthetase can distinguish between the two is again the interaction that the side chain makes with the microenvironment of the active site. An isoleucine R group will definitely have interactions that are going to lead to free energy changes that are more favorable than interactions made by valine side group. The delta G that will be there for the will be less negative than this. So that is one way of distinguishing. But still, because of its small size, suppose for example, isoleucyl tRNA synthetase accommodates valine and adenylates it. So then what does it mean? It means that an adenylated valine is now present inside the active site of isoleucyl tRNA synthetase. Now what? So such tRNA synthetases or enzymes have what is called as an editing pocket. So the editing pocket, okay, is large enough to hold valine, but it is not large enough to allow isoleucine to come in. Again, what is taken advantage of is the size, space, the spatial arrangement. So therefore, what is going to happen is the valine, adenylated valine will enter the editing pocket, and once it enters the editing pocket, it is going to get hydrolyzed. But the isoleucyl once adenylated and it's present in the active site cannot enter the editing pocket and will not get hydrolyzed. So that's how the isoleucyl tRNA synthetase, because of the presence of an editing pocket, is able to proofread the reaction that it has carried out. If it has made a mistake, then it can correct it by hydrolyzing the wrong one through the editing pocket. So if you look at the structure again, you will find that this catalytic domain is close to the CP1 domain. This CP1 domain forms the editing pocket where the adenylated valine can enter and get hydrolyzed, but isoleucine cannot enter. So that is how the editing pocket of many amino acid tRNA synthetase is able to ensure that it is adding the correct amino acid, or it is activating the correct amino acid. So it is ensuring the first step is correct. Now, interesting observation is that most organisms, most prokaryotes would have 20 aminoacyl tRNA synthetase for each of its 20 amino acid. But it has been observed that many prokaryotes have lesser or fewer aminoacyl tRNA synthetase. So here we will explain with an example. Now, this tRNA is specifically glutamyl tRNA, which means that this glutamyl tRNA can bind to its cognate amino acid, which is glutamate. As you can look at over here, glutamate has two methylene groups with a side chain, which is the carboxyl group. So it has two carboxyl group, that's why it's glutamic acid. Now, in many bacteria, what has been observed is that uh, it is possible that this group, okay, is aminated. So the OH group of the COH can be converted to NH2. If that is the case, this is CONH2. Now, what does this mean? It means that this amino acid is no longer glutamic acid, but it is glutamine. But this tRNA that is present is still glutamyl tRNA and not that for glutamine. So because of the enzyme present that can aminate a glutamate into glutamine, the organism can live without the tRNA for glutamine because it has this enzyme that can actually enable a glutamate to get converted to glutamine when needed. So therefore, in such a case, one does not have 20 amino acids. One doesn't have 20 tRNAs, amino acyl tRNA synthetases. One has 19 amino acyl tRNA synthetases, and likewise. So this is again a very important, interesting fact. Now let us therefore conclude. Aminoacyl tRNA synthetases with its multiple domains are able to accurately charge the tRNA with its cognate amino acid. The enzymatic reaction involves a two-step reaction. One that arylates the amino acid, which is activation of the amino acid, and then transferring that activated amino acid to the 3' end of the tRNA. That is, the tRNA gets charged. The aminoacyl tRNA synthetases has to be highly efficient in distinguish the tRNAs and also recognizing the isoaccepting tRNAs, as there is only one dedicated enzyme for an amino acid. So therefore, this one dedicated enzyme will not just have to recognize the isoaccepting tRNAs, but would also have to recognize all the isoaccepting tRNAs for the same amino acid, not for different amino acids. The most important and significant responsibility of the enzyme is to efficiently add the correct amino acid to the tRNA, which it may be able to do by proofreading, through editing pocket, or by ensuring that the interactions lead to a free energy change that is favorable. Thus, most organisms have 20 amino acyl tRNA synthetases for each of its amino acids, with the onus of the enzyme being to ensure that tRNA as adapters are ready for the process of translation. Thank you.